There is an organ in your body that filters your entire blood supply roughly 300 times every single day. Not once, not a dozen times, 300. Every 24 hours, without pause, without rest, without a single conscious instruction from you, this organ processes approximately 45 gallons of fluid, reclaims nearly all of it, and discards only what the body cannot afford to keep. Most people live their entire lives without ever thinking about it. And yet, without it, the chemistry of life itself would collapse within days. The kidneys are not glamorous organs. They do not beat like the heart. They do not expand with breath like the lungs. They do not carry the cultural weight of the brain or the visceral intimacy of the gut. They sit quietly, one on each side of the spine, tucked behind the peritoneum in a space so protected it takes significant force to reach them. Fist-sized, bean-shaped, reddish-brown, and they are, in the most literal sense, the reason your blood remains chemically stable enough for any other organ in the body to function at all. Understanding the kidneys is not simply a matter of memorizing anatomy. It is a matter of understanding filtration at a scale and precision that no human-engineered system has ever fully replicated. Every hour, these two organs make thousands of microscopic decisions about what the body needs and what it must lose. They respond to hormones, to blood pressure, to hydration, to the salt content of your last meal, to whether you are sleeping or running, resting or stressed. They are not passive filters. They are active, adaptive, chemically intelligent structures, and the story of how they work is one of the most intricate narratives in all of human biology. But before any of that can be understood, there is a question worth sitting with. A question that quietly changes how every part of what follows should be heard. Why does the body need filtering at all? The answer begins with metabolism. Every cell in the body consumes energy, and consumption produces waste. Proteins are broken down and leave behind nitrogen-containing compounds, primarily urea, that are toxic at elevated concentrations. Cellular activity shifts the acid-base balance of the blood. Minerals enter the body through food and drink in amounts that fluctuate wildly from day to day. The concentration of potassium, sodium, phosphate, and bicarbonate in the blood must be held within ranges so narrow that even minor deviations cause symptoms ranging from muscle cramps to cardiac arrhythmia to death. There is no stable internal environment without continuous correction, and the kidneys are the primary instrument of that correction. The ancient Greeks were aware of the kidneys. They knew they were connected to urine, but they did not understand filtration in any meaningful sense, and for most of recorded history, the kidney was thought to be a passive structure, a kind of biological strainer that simply separated liquid from solid. It was not until the 17th and 18th centuries, as microscopy began to reveal the cellular architecture of tissues, that scientists started to appreciate that the kidney was something far more complex. And it was not until the 20th century, with the development of micropuncture techniques that allowed researchers to sample fluid at individual points along the filtering tubes, that the true elegance of renal physiology began to come into view. What they found was not a strainer. It was a factory. Each kidney contains approximately 1 million functional units called nephrons. A single nephron is small enough to be invisible to the naked eye, yet it performs a sequence of operations so sophisticated that replicating it artificially remains one of the great unsolved challenges of biomedical engineering. The dialysis machine, the most advanced human substitute for kidney function, manages to replicate roughly 10 to 15% of what a healthy kidney does. This is not a failure of technology so much as a testament to the biological complexity it is trying to replace. The nephron begins at a structure called the glomerulus. Picture a tiny knot of capillaries, blood vessels thinner than a hair, coiled together inside a cup-shaped chamber called Bowman's capsule. Blood arrives at the glomerulus under pressure, and this pressure drives a process called filtration. Water, salts, glucose, amino acids, urea, and other small molecules are forced out of the capillaries and into the capsule, forming a fluid called the filtrate. Large proteins and blood cells remain behind in the capillaries because the walls of the glomerular capillaries are selectively permeable. They allow small molecules through while retaining larger ones. This selectivity is not incidental. It is architectural. The capillary walls contain specialized cells called podocytes, which extend long foot-like projections that interdigitate with one another, creating filtration slits of precise dimensions. The geometry is the function. The rate at which the glomeruli produce filtrate is called the glomerular filtration rate. In a healthy adult, this rate is roughly 125 milliliters per minute, which amounts to about 45 gallons per day. This is the 300 times per day figure. Every drop of blood in the body passes through the kidneys and is filtered at this extraordinary rate. But here is the immediate problem. If the kidneys simply excreted all of that filtrate, the body would be drained of fluid in roughly 30 minutes. This is where the nephron reveals its true complexity. Beyond the glomerulus, the filtrate enters a long, folded tube, and it is along this tube that the kidney's most intricate work happens. The tube is divided into regions, each with distinct cellular architecture and distinct functions. The first region is the proximal convoluted tubule. Here, roughly 65 to 70% of the filtered water is reabsorbed, along with nearly all of the filtered glucose and amino acids and a large portion of the filtered sodium. This reabsorption is not passive. Cells lining the proximal tubule are densely packed with mitochondria, the energy-producing organelles of the cell, because the work of pumping molecules from the tubule back into the surrounding blood requires significant energy. The brush border, a dense carpet of microscopic projections on the inner surface of these cells, vastly increases the surface area available for transport. At this scale, geometry is once again doing the work. The filtrate then descends into a region called the loop of Henle. This loop dips deep into the inner region of the kidney, a zone called the medulla, and then rises back up. The descending and ascending limbs of this loop have opposite properties. The descending limb is permeable to water, but not to salt. The ascending limb is permeable to salt, but not to water. This asymmetry creates a concentration gradient in the surrounding tissue, a region of increasingly concentrated fluid in the deepest part of the kidney. This gradient is the key to the kidney's most critical ability, the ability to produce concentrated urine. When water is scarce, the body releases a hormone called antidiuretic hormone, or ADH, from the pituitary gland. ADH acts on the collecting duct, the final segment of the nephron, making it permeable to water. Because the collecting duct passes through the concentrated region created by the loop of Henle, water flows out of the duct by osmosis into the surrounding tissue and is reclaimed by the bloodstream. The result is small volumes of highly concentrated urine. When water is abundant and ADH levels are low, the collecting duct remains relatively impermeable to water, and more dilute urine is produced in larger volumes. The entire system is continuously modulated in response to the state of the body. This is one of the most elegant examples of feedback regulation in all of physiology. The hormone senses need. The tissue responds to the hormone. The output changes to match the need, and the process repeats continuously, adjusting in real time, with a sensitivity that allows the kidneys to maintain blood osmolarity within a range so narrow that deviations of just a few % are detected and corrected within minutes. But the kidney does not only regulate water, it regulates acid. The blood must be maintained at a pH between approximately 7.35 and 7.45. Below this range is a state called acidosis. Above it is alkalosis. Either direction, if sustained, disrupts enzyme function across nearly every system in the body. The kidneys manage blood pH by controlling the excretion of hydrogen ions and the reabsorption of bicarbonate, the blood's primary buffer. When the blood becomes too acidic, the kidneys excrete more hydrogen and retain more bicarbonate. When the blood is too alkaline, the process reverses. This acid-base regulation operates in parallel with water regulation and in coordination with the lungs, which adjust the concentration of carbon dioxide in the blood, another determinant of pH. The kidneys and lungs together constitute the two primary mechanisms by which the body maintains chemical equilibrium. The kidney also regulates blood pressure, and this is where its role extends far beyond simple filtration. Embedded in the walls of the afferent arterioles, the small blood vessels entering the glomeruli are specialized cells that detect changes in blood pressure. When pressure falls, these cells release an enzyme called renin. Renin initiates a cascade of chemical reactions that ultimately produce a molecule called angiotensin II, which constricts blood vessels throughout the body, raising blood pressure. Angiotensin II also stimulates the release of a hormone called aldosterone from the adrenal glands, which sit just above each kidney. Aldosterone causes the kidneys to retain more sodium, and sodium retention draws water with it, increasing blood volume and, in turn, blood pressure. This entire system, called the renin-angiotensin-aldosterone system, is one of the most studied and most pharmacologically targeted pathways in all of medicine. Many of the most commonly prescribed medications for high blood pressure work by interrupting one or more steps in this cascade. There is another hormone the kidneys produce, one that connects renal physiology to a completely different organ system. Erythropoietin is a hormone released by the kidneys in response to low oxygen levels in the blood. It acts on the bone marrow, stimulating the production of red blood cells. This is why chronic kidney disease so often causes anemia. When the kidneys are damaged, erythropoietin production falls, red blood cell production declines, and the capacity of the blood to carry oxygen is reduced. The kidneys, it turns out, are not just filtration organs, they are endocrine organs. They produce hormones that regulate processes in tissues, far from the kidney itself. The kidneys also play a central role in calcium and bone metabolism through their activation of vitamin D. Vitamin D, as it enters the body through diet or is synthesized in the skin through sun exposure, is not yet biologically active. It undergoes a first transformation in the liver and then a second, critical transformation in the kidney, where it is converted into its active form, calcitriol. Calcitriol regulates the absorption of calcium from the intestine and plays a key role in maintaining bone density. Patients with advanced kidney disease frequently develop bone disease for this reason. Because the activation of vitamin D fails, calcium absorption drops, and the parathyroid glands begin drawing calcium from the bones to maintain blood levels. A single organ, water balance, acid-base regulation, blood pressure, red blood cell production, calcium metabolism. The list accumulates with something approaching quiet astonishment. Looking at the kidney from the outside, the anatomy is deceptively simple. Each kidney is protected by 3 layers. The innermost is the renal capsule, a tough fibrous membrane directly encasing the organ. Surrounding this is a layer of perirenal fat, a fatty cushion that protects the kidney from impact and helps hold it in position. The outermost layer is the renal fascia, a sheet of connective tissue that anchors the organ to the surrounding structures. The combination of these layers explains why the kidneys are so well protected. They are tucked behind the peritoneum, cushioned in fat, anchored in fascia, and shielded from the front by the abdominal organs and from the back by the thick muscles and bones of the lumbar region. Significant trauma is required to injure a kidney that is not already diseased. Sliced open, the kidney reveals 2 distinct internal zones. The outer region, called the cortex, is paler and appears granular. This is where the glomeruli and the proximal and distal convoluted tubules are located. The inner region, called the medulla, is darker and is arranged in cone-shaped structures called renal pyramids. The tips of these pyramids, called papillae, point inward toward collecting spaces called the renal calyces, which merge to form the renal pelvis. the funnel-shaped chamber from which urine exits the kidney through the ureter toward the bladder. The blood supply of the kidney is extraordinary in both volume and architecture. The renal artery, branching directly from the aorta, delivers approximately 20% of the heart's total output to the kidneys at every beat. This is a disproportionate share for two organs that together weigh less than 1 pound. But volume is essential to filtration. The glomeruli need pressure to drive filtration, and maintaining that pressure requires a constant, high-volume blood supply. The blood exits the kidney through the renal vein, returning to the inferior vena cava, with its composition already adjusted, its waste products extracted, its fluid volume recalibrated. It is worth pausing on what this means in practical terms. Every time the heart beats, roughly a fifth of all the blood it pumps passes through the kidneys. Every minute of your life, more than a quart of blood is being processed, filtered, and returned. For every hour spent in any activity, every chemical in that blood is being monitored and adjusted. The kidney is not a background organ. It is, in terms of its continuous workload relative to its size, one of the most metabolically demanding tissues in the body. The renal cortex, where most active transport occurs, consumes oxygen at a rate comparable to the heart muscle. And yet the kidneys have a remarkable capacity to compensate for damage. Because each kidney contains approximately 1 million nephrons, and because the body rarely needs all of them operating at full capacity under normal conditions, significant nephron loss can occur before any symptoms appear. Studies indicate that a person can function with as little as 20 to 30% of total nephron mass before kidney disease becomes clinically apparent. This reserve capacity explains why a person can donate one kidney and live a largely normal life. It also explains why kidney disease is often silent until it has progressed considerably, which is one reason that conditions such as hypertension and diabetes, both of which damage the glomerular capillaries over time, can cause significant renal damage before it is detected. The microscopic damage done to glomeruli by sustained high blood pressure is among the most important and least appreciated mechanisms of kidney disease. Under chronic pressure, the capillary walls thicken, the filtration slits narrow, the podocytes become stressed and drop away. The glomerulus scars. This process, called glomerulosclerosis, is irreversible. Once a glomerulus scars, the nephron it served is lost permanently. The remaining nephrons compensate by working harder, which increases the pressure on their own glomeruli, accelerating further damage. This cycle of compensation and injury is one of the reasons that chronic kidney disease tends to be progressive, self-sustaining, and ultimately difficult to halt once it reaches a certain stage. Research continues to explore the mechanisms by which podocyte stress leads to glomerular scarring, and there is significant interest in identifying therapeutic targets that might slow or interrupt this process. Evidence suggests that certain classes of medications originally developed for diabetes have protective effects on the kidney independent of their glucose-lowering action. Though the precise mechanisms are still being investigated, there is still no full consensus on the optimal strategies for slowing the progression of chronic kidney disease across different patient populations, and this remains one of the active frontiers of nephrology. The kidneys also present an interesting case study in how scientists come to understand an organ. For much of history, the kidney was studied at the organ and tissue level. Gross anatomy described its location and external features. Histology revealed the tubular architecture. It was not until the 20th century that the physiology of individual nephron segments began to be understood through innovations like micropuncture, which allowed researchers to insert tiny pipettes into individual tubular segments in living animals and sample the fluid inside. What they found repeatedly violated expectations. Molecules were being secreted into the tubule, not just reabsorbed from it. The kidney was not just reclaiming what it had filtered. It was actively adding waste products to the filtrate from the blood, a process called tubular secretion that increases the efficiency of waste elimination without requiring additional filtration pressure. This finding alone reshaped the entire conceptual model of renal physiology. Today, the understanding of the kidney has extended into the molecular level. The specific transport proteins embedded in the membranes of tubular cells have been identified, cloned, and characterized. Mutations in many of these proteins are now known to cause specific inherited kidney disorders. The signaling pathways that respond to ADH, to aldosterone, to parathyroid hormone, to the oxygen-sensing mechanisms that trigger erythropoietin release have been mapped with increasing resolution. The kidney has become a model system for understanding how epithelial cells, cells that line tubes and surfaces throughout the body, carry out vectorial transport, the movement of molecules in a specific direction across a cell layer. But perhaps the most profound thing about the kidney is what it reveals about the scale of biological precision that sustains a human life. The margins within which the body operates are remarkably narrow. Blood potassium must be held between approximately 3.5 and 5 milliequivalents per liter. Blood sodium between approximately 135 and 145 milliequivalents per liter. Blood pH between 7.35 and 7.45. Blood osmolarity between approximately 280 and 295 milliosmoles per kilogram. These are not rough guidelines. They are hard constraints maintained continuously against constant perturbation from diet, activity, temperature, stress, and disease. The kidney maintains them without ever stopping, without rest, without reset, through sleep and waking, through feast and famine, through heat and cold, through the chemical noise of an entire lifetime of metabolism. It adjusts without instruction, responding to molecular signals on timescales of minutes, holding the internal environment in a state of dynamic stability that the rest of biology depends upon entirely. There is a phrase sometimes used in physiology: the constancy of the internal environment is the condition of the free life. The French physiologist Claude Bernard wrote something close to this in the 19th century, long before the mechanisms were understood. He had identified something true simply by observing what happened when that constancy was disrupted. Without stable internal chemistry, no complex biological process can proceed reliably. The kidney is the primary guardian of that stability. Not the only one, but the primary one. When you consider the kidney not as a filter but as a regulator, its significance changes completely. It is not there to clean the blood in the way a water filter cleans water. It is there to hold the blood in the precise chemical state that allows the heart to beat with the right ions, the muscles to contract with the right calcium gradient, the brain to function with the right electrolyte balance, the enzymes throughout the body to operate within their functional range. Every organ system in the body depends, in some way, on what the kidney does. The kidney does not serve the body. In a very real sense, it enables the body to exist at all, fist-sized, quiet, hidden, operating at the edge of what biology is capable of producing. The kidneys are, without exaggeration, among the most sophisticated structures that evolution has ever constructed. And they are, right now, at this moment, filtering. If this is the kind of science that makes you see your own body differently, hit the like button and subscribe to Anatomy Unlocked, where every organ has a story this deep and none of them are done telling it.